Greetings, everyone. On behalf of the GeneBank platform and the Crop Trust, thank you for joining us for today's Grow webinar. I'm Yanni Van Beem, and with me is Faye Wambua Ludeling, who is co-moderator for this presentation. I'm very pleased to introduce our speaker today. Dr. Colin Khoury directs the Department of Science and Conservation at the San Diego Botanic Gardens and is a visiting researcher at the Alliance of Biodiversity International in Seattle, where he has worked for the past decade. He received his PhD from Wageningen University and also previously worked at the Crop Trust and USDA. He spent the last 2 years reviewing change over time in crop diversity worldwide, a project at St. Louis University funded by USDA's National Institute of Food and Agriculture. The title of his presentation today is Crop Genetic Erosion, a review of 100 years of evidence, thoughts on future research, and an outline of steps needed to mitigate, stem, and reverse further losses. Welcome, Colin.
Thanks very much. Good morning, good afternoon, good evening to you all. Thanks for your flexibility on the timing for me. I realize this is a bit of a different timing. That's because I'm here in San Diego, California, which is a little bit early. We're going to watch the sun come up together. So thanks for the opportunity to share our research with you, research that's been going on for the last couple of years and builds on many more years of research. There's a paper that the link was put into the chat already that this is associated with that just was published yesterday. Happy to see it out. Wanted to specifically mention to those in this community, which I assume are many of you in the ex situ conservation community, that in the supplementary materials are most of the results focused on change over time in ex situ facilities. And so those sections might be particularly interesting to you. To start this presentation, I'd like to give you just a little bit of story of my background and how I got interested in this particular question. I grew up with the narrative of loss of biodiversity for sure since I was young, taught that in school. I didn't particularly question it and definitely experienced it myself in terms of wild plants around me in Southern California, where I grew up. As I moved up mostly in the ex situ community, I eventually arrived at the Crop Trust itself, the organizers of this event. And at that time, I was working somewhat for the director, helping the director communicate these big messages around crop diversity and its loss to folks around the world. And the director asked me to come up with numbers, to come up with statistics and visuals to communicate these messages. And so I started digging into some of the major themes or memes, if you might call them that, around our field. And one of those, maybe the most dominant of all, is the 75% narrative. There's a bit of text on this slide. This just happens to be the last time that I saw this 75% narrative in the media or in a paper in this case. It was this last week. And this is a paper put out by an educational university in Vermont in the United States. And in blue, you can see this very classic quote. Agricultural biodiversity throughout the world has experienced a sharp decline, with around 75% of crop genetic diversity being lost. I would guess that just about everyone in this crowd attending this has seen this number at some point or another. If you look around, you'll start to see it just about everywhere. It's fascinating to me because it's repeated an untold number of times in published literature and also in the media. And yet there might be no number more inflammatory, creating more anger and ire within this community than this number. And that's been a fascinating thing to me. And so one of the starting points for my research over many years now has been digging into numbers like this and trying to understand what the truth is behind them, how accurate they are. One very peculiar thing about this number itself is that it's incredibly difficult to find the source of it. So you can see perhaps on the slide that there is a citation there. It says number 1. That citation goes to an authoritative source, which in this case is the website of the Convention on Biological Diversity, a page called What's the Problem? And down there in that page, here's the quote, which is cited. According to the FAO, it is estimated that about 3/4 of the genetic diversity found in agricultural crops has been lost over the last century, and this genetic erosion continues. So there's a citation, but you might note that the quote itself has no citation, has no source. And this is where we invariably end is saying that the FAO has said this, but no way to know when, how, and from what evidence base the FAO has said this. I'll talk a little bit more about this story a little bit later. We tried to dip in and figure out the actual answer of the source, if you hang on towards the end of the presentation. So as mentioned, I spent the last couple of years, I was really lucky enough to spend really thinking about this issue and being inspired by those who have tried to approach this question from an evidence base, in particular, revisiting sites that were documented in previous decades or previous years, like this work by Gary Nabhan with Hopi community here in Arizona in the United States, and going back to the same farmers or farming families or communities and having conversations with those families about what has changed over time. The last 2 years, I was really supposed to be in the field doing the same sort of work, especially in Peru and in Mexico. Because of COVID, that was shifted quite a lot and I wasn't able to travel. And so I turned around and worked much more on this review of the entire state of knowledge across the world about what we know of this change over time. This comprehensive sort of review had really not been done before, although there had been a number of different sorts of reviews. Yongbi Fu in 2006 had done a review of 23 articles using genetics at that time, rapids and AFLPs and SSRs, basically on wheat and maize and barley, and looking at change in modern cultivars. Van de Wouw's seminal work in 2009 and 2010, especially the 2010 meta-analysis, again on modern cultivars and looking at change over time. There was a review by Ralph et al. In 2010, unclear what methods were used exactly in that one or how many papers were put in, but an extensive review. And then more recently in 2015, there was a book chapter by Thorman and Ingalls that tried to cover or provide an overview of this issue, but no real comprehensive attempt to look at all of the evidence across the whole world. Having done this review over the last couple of years, I now understand in part why it hadn't been done before, perhaps. It was among the most challenging scholarship tasks of my career so far, perhaps in part because we really tried to understand this big picture, which is endless and not entirely documented, as you will see. So what do we do here? Let me start by just a couple of definitions to let you know our scope here. When we were looking at crop diversity, we were really looking at the full extent. So not just at the genetic level, but at species level, at varietal levels, and within varieties or infraspecific levels. Therefore, our definition of genetic erosion, which by the way has been defined really an untold number of times itself. We found at least 50 definitions, all different in the literature. We defined it quite widely, just trying to understand loss in this diversity at any of these levels in terms of species, varieties, and genetic or phenotypic diversity, the whole picture. So further on our scope, we tried to understand this change across all crops and all their wild relatives. I would like to note that this term genetic erosion has gone beyond our field and is widely used now in wild biodiversity. And so there's easily 400 articles with this term genetic erosion in the title now, and maybe 23,000 articles with that phrase in the text. So it's much bigger than us at this point, if we're talking about crop diversity conservationists. We looked at all time periods that we could find from as far back as possible to the present, all locations around the world, all geographic scales. To explain that, there are articles that really try to cover sort of a global picture for a specific crop or something like that. There's all the way down to looking at a change in one species or change in one accession in one gene bank somewhere. And so that whole entire scale. All taxonomic scales, as I mentioned, the main ones that most folks have focused on are at the species level, at the variety or population level, or at the gene level, let's say, or the trait level. All analytical methods. That involves really a wide range of work that people have done around the world, a lot of field work and surveys, that is talking with farmers and trying to understand change from their perspective, certainly genetic analyses, phenotypic or morphological analyses, literature-based comparisons, modeling, everything we could find. And all targets of understanding change. There are also three major ways that folks have mostly approached this, or mixtures thereof. There's absolute loss, for instance, of a species or of a variety. There's changes in the numbers in a particular place, like in a community of varieties being grown, and there's changes in the abundance or frequencies within a particular place. One aspect that I also wanted to mention is that there is a wide spectrum of what I would consider to be called direct analyses in this sphere, or indirect analyses. This is an example that we present in the paper of a sophisticated, relatively direct analysis by Thorman et al. In two papers around 2017, where they revisited sites that had originally been collected in 1981 of both cultivated and wild barley. We went back to the exact same locations and collected again. They then grew those same samples from both time periods in a common garden, and then performed phenotypic and genotypic analyses on them. There's also a wide range on the other side of that comparison spectrum, indirect. For instance, the well talked about work by Fowler and Mooney, where they looked at seed lists from the United States of vegetables and fruits that were in commercial seed listings in 1903 in the United States, and compared that list to gene bank accessions in the National Seed Bank here in the United States from 1983, and tried to make a comparison between those two data sets of the diversity there. All right, so here's a summary of what we've found before I go into each specific topic that might be particularly of interest to you. Looking across all the literature we could find, and again, really just focusing on change over time and evidence thereof. So not a general statement, but a specific evidence-based statement. We found around 300 or 288 articles that we felt could have evidence about crop diversity change, 232 of which had primary data to which, and then we focused on particularly on those. To do this, we used essentially a snowball methodology, which is that we read everything that we could on this subject and then kept following the citations. We also did searches online across literature databases, including Google Scholar and others, mostly in English, but also in Spanish, Portuguese, and French to some degree. So these papers were published between 1939 and 2021 in the present, and I thought you might be interested in some of the journals that have the most of these papers. Genetic Resources and Crop Evolution within our community being the one that has published the most. These are the crops that you will see. The numbers after each of these lines and statistics are the number of publications. So out of these 232, so there are 50 papers within that have looked at wheat. Now, a meta-analysis of this wide range of papers around the world or articles was really not possible. There's an incredible variety of methods and taxonomic scales of ways and approaches, and very little actual available data underneath it. That is the primary data. And so we really provide these summary numbers as a sense of what we know around the world, but it shouldn't be considered a statistically significant data set really in terms of a proper meta-analysis. The scale at which these analyses were performed were often within country, but across many communities. So we called that sub-country, also at the country level, regional level, global level, or smaller levels like the community. And the time periods in which these papers focused were mostly from the 1900s to the 2000s as the starting point, up to the 1990s to the 2010s as the ending point. There's a wide range there, and that's why you see some overlap there. In the parentheses, you see the very earliest time period. That might be surprising for you, 4,000 years before the present. That's because of new techniques in archaeogenomics that allow studies across very long time periods, and then modeling up to 2099. Median length of the studies was about 40 years. And as I mentioned, studying within varieties, varietal levels, and species level diversity. The tools used, we touched on a little bit before, but a lot of genetic studies, a lot of these field studies or surveys, talking with farmers, nomenclatural studies, which are basically these desk-based comparisons based on lists, pedigrees, that sort of thing, or pedigrees are listed there too. Phenotypic studies, modeling, using information mostly from the seeds themselves, biological materials, from farmer knowledge, other published information, field observations, and others. So that's our overview essentially of this whole data set and what we learned. To organize this and to come to a sense of how we could talk about it, we ended up with 5 environments or contexts. The first one is change in traditional cropland races on farms. The second is changes in modern crop cultivars. The third is crop wild relatives. The fourth, which again might be the most interesting to this crowd, is change within gene banks or in ex situ conservation facilities. And the fifth was change essentially in human diets or food supplies and food systems. All right. So I'm going to take a deep breath right now, and I invite you to do the same if you will. Hopefully you have some tea or coffee with you. This is a dense presentation. It's got a lot of information to it, and I hope will be interesting to you. I'm going to try to work pretty fast because there's a lot to say, and I'd certainly love to answer questions or have discussions with you more than anything. And so let's take a deep breath. And chapter 1, cropland races on farms. So 139 articles published throughout our entire data set from 1940 to the present. This research based on traditional land races is mainly on annual cereal crops: maize, wheat, rice, barley, and sorghum. There is relatively broad geographic coverage globally, with particular focus on East Africa, on Mesoamerica, West Africa, South America, South Asia, and Southwest Europe, especially in Ethiopia, in Mexico, Italy, USA, Peru, India, Philippines, and a few other countries. Regarding the scale, mostly it's at this sub-country scale across a number of communities. These studies are mostly studying at the varietal level, or that is change in diversity among land races, but also include change within land races and species level diversity to a smaller degree. Mostly using these social methods, talking with farmers or doing field surveys, but also genetics and other analyses. And thus mostly based on farmer knowledge, but also on the biological materials themselves. And this study period, again, most of the papers were studying from around the 1920s to the 2000s up until the 1990s to 2010s, about 30 years on average. All right, so what did we find? So this literature base documents widespread losses of landrace diversity over the past century, continuing to the present. Over 96% of the studies found change in diversity over time, with about 86% documenting loss. These include the complete disappearance of specific land races and a few crop species, also declines in richness, the numbers of land races being grown, and losses of within landrace variation. There's proxies for those, including changes in harvested area or the number of farmers cultivating specific land races. There's also an interesting trend here. I certainly didn't expect to find it, and I'll come back to this a couple of times, which is not only losses or changes in numbers, but also increasing similarities in the remnant land races that are there. In terms of drivers or reasons for change over time, the most commonly reported driver in the literature is exactly what has been talked about since Erna Bennett 50 years ago, which is the replacement of land races by modern cultivars. That said, there are many, many, many more drivers that are now recorded, more reasons for change over time. This is a figure that we produced in the paper that just tries to contextualize some of those drivers within an evolutionary forces or evolutionary kind of drivers context, and looks at how this all kind of comes together. The real answer is that it's very complicated. On the other hand, it's not totally random. These drivers are essentially interrelated facets for the most part of agricultural and economic change, manifested through extension and expansion of formal seed systems, globalization of markets, increasing availability of agricultural technologies and policies that support those processes. There's an evidence base that says that there are certain conditions that lead to these changes. In particular, for instance, farmlands with characteristics that are amenable to modern cultivars, for instance, flatter and irrigated plots rather than mountainous, topographically diverse areas. Major changes in labor availability and demographic changes, that is people moving to the city. Demand and market changes, and periods of instability, such as war and civil strife, but also environmental change. All right, so far we see that there really is quite an evidence base that there's been a tremendous amount of loss and amount of change in land races. That said, there are a number of caveats that have to be mentioned. This is these are cartoons that we found that were produced in the 1960s and 1970s by the University of Sydney. An incredible set of scientific cartoons that were produced in Australia, New Zealand, in the United States and Europe, and across the world. They were reproduced in newspapers. And this was one from 1971 called Genetic Conservation. And there's a figure in our paper that shows this series of 5 slides across 5 days that were produced and sent around the world. The very first part of the text here on part 3, which would have been the Wednesday of that week, says, the primitive strains of wheat, peas, and beans grown in the Middle East have not changed substantially in thousands of years. This is one of the caveats that I wanted to mention, and that is that, in fact, as studies have become more sophisticated over time, they have certainly realized that interchange and turnover of land races is, in fact, much more common. These populations of evolving diversity are certainly always evolving and changing. And so not change substantially is really an untruth. Second point is that losses in this genetic diversity that have been mentioned have been shown to be much less dramatic, or more gradual at least, in many other crops besides the staple cereals, especially woody perennials like apples and grapes, but also common beans and even in sorghum. More generally, basically, crops that aren't major commodity crops with a lot of formal plant breeding are certainly less exposed to this loss in land races. Now, this loss in particular genotypes doesn't necessarily mean some total loss. Basically, it's much more difficult to account for what's been replaced or what's come in than it is to just simply mark what has been lost. A couple of examples of this that I think are interesting just to ground this down. So there was a paper in 2009 by Steele et al. That found that replacement of rice land races by modern cultivars in Nepal could actually increase genetic diversity in a given region, as long as the adoption of modern cultivars was limited to 65% of the study area. And Vigouroux et al. In 2011 in Niger found basically no major change in genetic diversity over 25 years in pearl millet, even with major changes in climate and recurrent drought. Finally, adoption of modern cultivars doesn't always particularly mean loss of land races. And often farmers will keep both of them or mix them together in a process in Latin America called creolization to arrive at something farmer bred that has the best of both worlds. So here's Friday in the same set. And I'd ask you to look at the third box there, which says, unfortunately, modern agriculture is spreading so rapidly in these areas that within 5 years of 1971, the primitive strains will have vanished. And this was, as I understand it through the reading, really the sentiment at that time period, that urgency, which started this massive amount of collecting around the world through international gene bank collections and national partners, and which really continues to this day. This is also, turns out to be perhaps a little more drastic than is the truth. In reality, there has actually been persistence of land races in many, many places around the world. The continued amount of publications around the world on land races essentially shows that there are still land races around the world. And so this prediction of genetic wipeout, as Jack Harlan said, also around that time, it turns out to be less of a wipeout than once thought. What is this about? Well, there are these distinct values that modern cultivars don't seem to be able to provide, certainly in all places around the world. They do provide in these contexts local productivity, stability in production, resilience. Essentially, they provide option value and risk management, especially to small scale farmers lacking the economic resources and credit opportunities and extension support. All right, so that was chapter 1. Now we're moving into chapter 2, which is modern cultivars. Okay, what do we know about modern cultivars? This story is more complicated than the last story. There's almost as much literature there. There's over 100 articles between 1984 published and 2021. These were also really on the annual cereals: wheat, barley, maize, rice, and oats. The geographic focus has been much less even across the world than for land races. Essentially, we know about Europe and North America, to a more limited extent, South and East Asia, couple other places, essentially the United States and Canada, France, the UK, China, India, the Russian Federation, and a few other places. Most of these studies have been conducted at the country or subcountry level. They're mostly looking at changes in within varietal or varietal diversity, that is of these modern cultivars, using mostly genetic methods, using the biological materials. All of these numbers, by the way, are in the paper, but I should give you a little bit of an overview. So what do we know about modern cultivars? All right, so essentially the whole data set does document change over time in these cultivars, and over 2/3 have shown loss or decline in diversity over time. Again, we find increasing homogeneity. That is, within the set of modern cultivars in a certain area, they're coming to be more similar at the genetic level through plant breeding in this case. All that said, many of the papers, in fact, well, close to 50%, also found, in terms of something they were measuring within the paper, some aspect of maintenance of diversity, and 50% also found increases in diversity over time. Let me just stop for a minute and say that many papers would find more than one finding. So at one level, they might find loss, but at another level or aspect they were looking at in terms of other traits or in terms of another analytical tool that you were using, they found increases or maintenance. These papers are not always simple, in many cases, not simple at all. So to provide a little bit of context for all of the different ways that this can go for modern cultivars, in the very first one that you see there in box A, and this is again a figure from our paper, this is more of the typical story of decline. This is a paper by Fu and Dong in 2015, looking at wheat cultivars in Canada, about 75 wheat cultivars in 3 different methods, and showing that across decades, as sets of cultivars, the diversity at SSR and levels have essentially declined. On the other hand, in the seminal set of studies by Van de Waal around 2010, that meta-analysis that I mentioned, looking at regional levels and looking at a different way of looking at it through allelic evenness rather than allelic diversity. That study showed that there was a drop around 50 years ago in diversity in modern cultivars, but since that time, the diversity has gone back up and through the 1990s, and essentially is evened out in the total data set. And so that that researcher was able to say that there was essentially no net loss of genetic diversity over time. A couple other caveats there that I thought you might find interesting. So another study by Fu in 2006, looking at 23 cultivar diversity publications, found something interesting, which was that genome-wide changes in genetic diversity were not significant over time, but allelic diversity at specific individual chromosome segments was quite substantial. Van de Waal in 2013 reported an increasing number and uniqueness of lettuce cultivars in Europe and in French and Dutch companies, but also a dramatic decline in the number of breeding companies. And then there was a recent study on rice cultivars in China that documented that same peak as you see in part B there in the 1990s of higher levels, but after that point, a decline in the diversity up until the present. That was Tang et al. In 2021 more recently. Okay, so part C there, here's an important point. So while significant changes in modern cultivars are certainly documented, determining the overall effect on functional diversity or on plant breeding is really pretty much a challenge. Most of the studies, the great majority, essentially are trying to understand overall or neutral diversity, and very few of these studies are able to focus on functional diversity or traits. Phenotypic studies, on the other hand, have tried to study functional traits, and most of those have showed loss or change, essentially loss over time. On the other hand, this particular paper we show here in section C, the colorful one there, this is by Schouten et al. In 2019, documented not only increasing genetic diversity over 7 decades in greenhouse tomatoes in Europe and the Netherlands, but also higher proportions of exotic intergressions from crop bio relatives since the 70s. To deal with pests and diseases, but also for organic systems and for the market demand for better tasting and different colors and different flavors and shapes of tomatoes. And so showing that plant breeding can also diversify systems when that's an objective. This is a heat map, by the way. It shows different chromosomal segments and a time period range from left to right there. Blue showing less diversity in terms of overall expected heterozygosity, and red showing higher amounts of diversity. And so you can see that over time, you're getting more heterozygosity at many chromosomal segments. Now, all that said, all of this data set, all of these data sets are still really just about available varieties, or what's out there, or what was out there in terms of cultivars. It's much, 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 much less common to find studies that show changes actually in cultivated areas, or really understand what's in the field across a region. This in section D, this is one exception. This was by Gatto et al. Also recently in 2021, showing essentially the varietal turnover from land races, which are shown in orange there, to modern cultivars across both sub-Saharan Africa and Asia, but especially in Asia, this major transition going on. And this has been shown to particularly happen through the spread of these mega cultivars that are certainly less genetically diverse as a whole than what they replaced. All right, moving on to chapter 3, crop wild relatives, more in my wheelhouse and my background. Crop wild relatives. So less papers have studied change over time in crop wild relatives. That data set is from about 1988 to the present, 33 papers. Essentially, we've studied the wild relatives of rice, of maize, coffee, barley, a few other things. There's pretty good regional spread, especially in East Africa and Mesoamerica, East and West Asia, West Africa, and North America, particularly in Mexico, China, Ethiopia, Kenya, Tanzania, a few other places. These studies mainly assess changes in diversity at the subcountry scale, that is again across a set of areas within a country, but also at the country scale. Many studies all the way at the species level, but also smaller levels within populations or population levels. A lot of field surveys, some genetic analyses, and some others. So what does the crop wild relative literature tell us? Not good, essentially. The worst among all of these stories. So 97% of the articles finding change over time, 90%, 9 out of 10, finding loss, sometimes severe loss. And also interestingly, once again, increasing homogeneity in the remnant populations. Let me break this down just for a minute. So a number of studies finding that through habitat disruption or disturbance, there would be increasing interflow, genetic flow between crop relative populations or with crops. And that would, in some cases, lead to increasing diversity actually in those populations, but also increasing similarity between those populations. So once again, for the third time, finding this homogeneity story. It's quite interesting. All that said, again, finding some maintenance of diversity over time and also some increases in diversity, essentially what I just mentioned about the increased gene flow. And the drivers are really what I think are known and expected to be habitat disruption, changes in, that is, in how land is used, also climate change, already recorded and certainly modeled in the future to impact crop relative populations quite a lot. And agronomic practices, that is essentially a change from traditional agronomic practices into modern cultivar practices, especially with mechanization that would remove crop relatives that are occurring within or nearby fields. And finally, we get to chapter 4, which is changes in ex situ facilities, that is changes in the diversity of crops within gene banks. About 30 articles have looked at this, published between 95 and 2021, mostly on rice and wheat and barley, a few other things, mostly on cultivated materials, but a little on crop relatives, and mostly essentially in North America and Europe and a few other places. A lot of these essentially use genetic or phenotypic comparisons to assess diversity kind of at the within variety or the variety levels to look at change over time. And so this is the results in the data set that you'll see in the supplementary materials if you're interested there. Every single one of those articles did document some change in that diversity in gene banks over time, with about 85% of them documenting loss or decline over time. That said, around 42% also found maintenance of some aspect of that diversity, and 30% documenting increase in diversity. In this case, you might notice that, or note that increase in diversity isn't particularly a good thing in this case, if what we're trying to do is essentially keep accessions the same. The main reported drivers in these data sets essentially around the regeneration and multiplication activities and also human error. We provide a figure also in the paper that looks at some of these changes and examines them. In that figure A, which I won't go into too much, this is a model decline just through regenerations, both in terms of wild and domesticated accessions over time, just with a regeneration essentially. That second paper is a quite interesting one. It's from a set of papers. It's about half the data set. That's really challenging because what it does is it tries to compare diversity in ex situ facilities to diversity in situ and make some comparison based on that. And that's that's challenging because in fact both of those contexts change a lot over time, especially the in situ side. But this very interesting article by Parzies et al. Way back in 2000 essentially looked at genetic diversity loss for Syrian barley land races stored for 10, as you can see there, 40 and 72 years throughout regenerations, and looking at and comparing to the diversity in existing in situ populations, and seeing that essentially the ones that have been stored the longest have now the least amount of diversity in comparison. It's an interesting set of papers. I think there's still a lot to be done there to really understand change. Happy to talk about that, but I'd invite you to take a look at the results that we find. I hope that they're at least thought-provoking. All right, so we're now going to head into the second from the last section here, which is noting that although I think this is the largest review that has ever been performed on understanding change in crop diversity over time, it still, I think, really only tells a tiny slice of the global story. And that is really because that evidence base still isn't out there, even after 50 years. Some have said that this is because the urgency of loss was so high that we've spent more time actually doing the work, doing the collecting, than just trying to analyze the change. And that makes great sense to me. That doesn't have to be argued to me, but it does mean that we don't quite know still the whole story where we're at now after more than 50 years, really 100 years of change. And so in the paper, we've tried to organize some thoughts about what are the major areas where we still need to work to understand better the state of change and diversity in crops. The first of those is breadth and complexity and inclusiveness of the research. When it comes down to it, very little is still known about changes in diversity in pulses, starchy roots and tubers, and vegetables, fruits, oil crops, sugar crops, much less forages and feed crops, fibers, medicinals, everything else besides the major cereal staples. While it might be, some of this knowledge might be transferable to cereals to others, there is definitely an evidence base that says and shows that other types of crops with other biological systems, especially, may have gone through different processes. For instance, may not have lost as much diversity as the cereals have. All this said, we also know very little about crop wild relatives still, especially about any of these other crops. There's very large areas on every cultivated continent that are still not really studied, and that includes the primary regions, the centers of origin of diversity, even for crops like wheat in the Fertile Crescent or maize in the Andes or in sub-Saharan Africa, many other places, potato in Europe. They really still haven't been studied in terms of this change over time. In terms of study scale, we know a lot about sort of within a couple of communities or at a seed system level, for instance, in Canada, about oat cultivars. What needs to happen in part is an expansion of those community level scales to the whole landscape from which we understand that seed diversity flows and through informal seed systems. And that often is across a region of a country, up to a whole country or bigger. There was a really beautiful set of papers, analyses or PhD work by McLean Rodriguez et al. In the last couple of years that I'd highly invite you to look at. We reproduce a little bit of her work also in a figure in this paper in figure 3 that looks at maize. Elegant studies at that scale where you can really understand most of where seed diversity is flowing among communities and among farmers. All that said, we also need better studies at scales which are largely ignored entirely in these analyses. For instance, in backyard gardens, which in terms of integrating studies where you really understand across a region, things like what's being grown behind a house are very, very rarely part of that. We know a little bit about crops, but we know even less about other forms of agricultural diversity like livestock and pollinators, soil microorganisms, and all of that. Ideally, we'll begin to do these analyses where we're integrating not only our understanding of crop diversity change in those places, but these other aspects into a more holistic sort of understanding of crop diversity change or agrobiodiversity change. And finally, while the expertise of crop diversity researchers that have been doing this, trying to understand this, is wide, there's plant breeders and geneticists, anthropologists, all kinds of things going on, very, very rarely really are farmers really involved in a high degree in these processes. There is a lot of interviewing of farmers, but real participation in ways that are now understood, for instance, in the climate change community, as along this spectrum of really collaborative or collegial are not seen yet in our field. Essentially, all the papers that I saw or reviewed, one would consider farmers to be essentially on this spectrum of contractual. And so what we may be missing through that more active and equal engagement is really a deeper and truer understanding of not only a change of the drivers and the realities for farmers. In terms of the robustness of the methods and the underlying theory, we do have to acknowledge that our understanding of the past is always going to be limited by not knowing what has already disappeared. We have ancient DNA techniques and all of that stuff, but it's really very, it's indirect measurements that we're really seeing. What we really need are larger scale direct comparative methods. And there is and there is agitation towards setting up networks, for instance, of participatory observatories around the world with especially indigenous communities, where over time studies could be done again and again under the same methodologies and really come up with much more sophisticated and direct measurements. Even though in those contexts, we have to realize that understanding this change over time in dynamic systems is incredibly complicated. I don't want to criticize this community and all of these papers too strongly for many reasons, but one of which is that it is incredibly hard to make good comparisons here. Focusing in on what is really significant in terms of change, I think, is one of the key pieces there. And that really means multiple time points over large enough time scale to really be significant towards diversity in a certain area and how it flows. Methods themselves also need further analyses. Many times, very similar studies will come up with very different results still. As I mentioned before, we still know a lot more about neutral diversity or overall diversity rather than these functional traits. That is also a very difficult thing because, essentially we don't really understand genomes well enough to know all of the traits and how to study them, how to connect phenotype and genotype. That is coming along, certainly, and hopefully eventually we'll have that, but there are no papers right now that I have found that study change over time, both at the phenotypic and genotypic level in terms of functional traits of interest. We can talk more about that if you'd like. All that said, we also don't know what functional traits are going to be important in the future. And so these overall genomic analyses are certainly probably going to continue to be relevant. This trend that I mentioned, not only in modern cultivars, but also land races and crop wild relatives of increasing homogeneity, is an interesting one. And I think we need to understand more about that, especially because if you're also understanding, for instance, cultivar turnover and pesticide use and things like that, it might be able to be a way to understand better where we're at with crop genetic vulnerability at seed system or landscape scales. And as also mentioned before, we still need to do a lot, not only to understand essentially the counts or what's available in terms of diversity out there, but also changes in what's actually been grown or the spatial areas, the harvested area of change. And that's where we really drill down again to understanding better, I think, genetic vulnerability or susceptibility based on uniformity. All right. You may remember that we had a 5th chapter, and that was trying to understand change over time in food systems and diets. It turns out that we didn't end up making it a chapter in the paper because there really isn't much evidence at all about change in crop diversity in human diets, essentially. This whole field, I think this whole part of the field really could use a lot more focus. It's also very technically challenging. For instance, still the questions around how much crop production diversity affects human dietary diversity on farms and subsistence farms is still a complicated thing. There's a marginally positive association there, but it's complex. This figure here is a paper from our work a number of years ago that did look at diversity in food systems across countries around the world and did find actually increasing diversity in terms of crop commodities around the world, around countries. But because of that increasing diversity of these mega commodities, increasing homogeneity across food systems around the world, moving from 50 years ago to the present. So not only in cultivars, not only in land races, not only in crop R relatives, but also in food systems, this trend of increasing homogeneity. Essentially what I want to say here, and I'm happy to talk more about this, is that there's a lot that we still need to know to connect crop diversity change to human diets and nutrition. We still really don't know much. And finally, the conservation implications. As I mentioned before, there was a lot of emphasis on action rather than theoretical analyses in the last 50 years, and that makes sense to me. There are papers and analyses that are really now trying to combine those studies, trying to understand change over time and then what we should do about it, and directly saying what we should do about it. I simply want to mention here that I think our target in the future should be to try to integrate these, particularly by being more holistic in our studies. That is, conceptualizing the total amount of diversity, for instance, across multiple crops in a given region or study area, and understanding what diversity is there in situ, what diversity is there ex situ, and how things are changing over time in the in situ systems particularly. Those methods have been proposed under a lot of names like early warning systems and conservation gap analyses and hotspot analyses, but they're really yet to be fully developed, particularly regarding the temporal change aspects. So I think we have a lot more to go there. So we're moving into the conclusions. So I'd like to go back to this oh so famous quote in our field. Since the beginning of the century, about 75% of the genetic diversity among agricultural crops has been lost. So we did some sort of forensic studies. We really tried to dig in and find where this came from, talked to a lot of people, looked at a lot of literature, kept digging and digging and digging. At this point, right now, the earliest that I have found of an actual citation for this is an FAO document from 1993 that says this. Now note, we don't know if this is the original document, but it doesn't seem like an adequate original document. And in fact, even in this original document, in another location, it says FAO estimates that since the beginning of the century, 75% has been lost. Perhaps indicating that FAO said this before, or maybe it's the first time and it's just the time where they say, we say this. But again, in both cases, there's no evidence provided. There's no background numbers behind that or anything. So what do we know about where this came from? I think the best guess at this point, drum roll please, is that it comes from the time period right before the Convention on Biological Diversity in the late 80s and into the 90s, and work by a number of different actors in the FAO at the Commission on Genetic Resources, in the IPCRI or IBPGR at that time, in non-governmental organizations such as the Rural Advancement Foundation International, RAFI, all these folks trying to essentially provide an evidence base that stakeholders, decision makers were asking for at that time of how much we've lost. And they were using anecdotal evidence, they were using their own experiences, they were building resources from people telling them what's happening around the world. And so how all that came together into 75% is really still a mystery, but that's the most likely case is essentially this kind of amalgamation of lots of pieces of evidence and number of people talking. Still don't know who said 75 first. On the other hand, it is possible that actually there is a singular source to this. And one real possibility for that comes back to Fowler and Mooney and Shattering in 1991. Both of them having worked at RAFI. And by the way, this paper in 1993 by FAO was written by a staff member of RAFI as well, Hope Shands. Anyway, in 1991, in Shattering, Fowler and Mooney remark on the feared loss of diversity in vegetable varieties in Europe due to changes in the allowable varieties on the varietal registration system, and the fear that up to 3/4 of traditional vegetable varieties disappearing. Where that came from for them was an interview that they did with Erna Bennett in the late 60s. At that time, Erna Bennett was working at FAO. And so if we understand that understanding of vegetable diversity in Europe got generalized into the 75% number and attributed to FAO via this conversation by Erna Bennett, then that is possibly one origin source to this number. All this should show that essentially, you know, it's not a clear number. It's not very accurate. We really still don't know exactly what it means. What do we mean, or what do we know? Well, I can't tell you how much has been lost over time. I apologize if you feel like you just wasted the last hour of your life, but we can report how many articles found losses in diversity, and about 80%, 79% of the articles documented loss. That's kind of close to the 75% in a certain way. But we also have to note that within our data set, 37% found maintenance of some aspect of diversity, and 30% found increases or appearance of new diversity. Despite not having made much progress really on understanding this single global number, we have had a great amount of progress in understanding change and its drivers. It's clear that enormous changes occurred. We know that enormous reductions in land races and farmers' fields and crop relatives in their natural habitats have happened and continue to happen. We also know that landrace diversity is still around and continues to be cultivated. There's these cycles that are seen in the literature for modern cultivars of loss and recovery. It's not clear where we're at right now. We really need another one of these meta-analyses that in the present of looking across major cultivars of cereals and hopefully a lot of other modern cultivar crop types. We know that increasing homogeneity among in seed systems, modern cultivars, even in land races and wild relatives is happening. And that is an interesting trend that may be connected eventually to genetic vulnerability, but we really need to study that more. And we know that change in diversity in ex situ systems is the rule rather than the exception. Much progress in understanding the conditions as well, like under what conditions is this happening? And so let me just read just a couple of, I think, some of the most important. Areas where connectivity around the world is rapidly increasing, either through habitat degradation or through new roads or through people moving around. All of these different things is where change is happening the most. Regions where agricultural communities are undergoing major demographic shifts in the older people passing on and younger people not continuing with agriculture, people moving countries, all of those things are rapidly changing crop diversity. Areas where climate change is the most acute, areas where there's war and civil strife, certainly, obviously. Areas where formal seed systems are losing public breeding institutions and are consolidating. And in conservation repositories that lack stable funding and adequate infrastructure. I'm sure that will make a lot of sense to you all. Now, what we don't know shouldn't keep us from not acting. I have a couple of final slides that are our thoughts about what we need to do. I think the ex situ side won't particularly be surprising to you. Hopefully it'll be, I think, mostly things that you've been saying a lot, but all of these things are very true through the evidence that we've acquired and what needs to happen. Given these ongoing losses in crop diversity in farmers' fields and natural habitats and seed systems, this simplification or homogenization trend that's happening in environments, increasing anthropogenic pressures, climate change, habitat destruction, caution only dictates that we need to continue to ramp up our crop diversity conservation ex situ. Nevertheless, the capacities of these repositories still in many cases do need bolstering and support to minimize genetic erosion in these environments. Safety duplication needs to continue to be a priority until it's essentially completed across the world. especially for crops that are not so difficult to safety duplicate, the non-seed crops. There is further work to be done to improve the accessibility of collections. I realize that this is a complicated subject and politically sensitive in some areas, but it is clear that gene banks are not as accessible as they could be to everyone, certainly. And it is also clear that there's new methods and innovations going on to more directly connect diversity to those that need it, even farmers directly. And there's some really interesting innovations going on there. Embracing non-conventional ex situ repositories simply acknowledges that it's not only the national gene banks and international gene banks out there doing this work, and that really looking across all of the different folks and trying to work together makes good sense. And that community seed banks, where there's a lot of evidence too, and certainly some challenges there in terms of their viability, but they're also performing very important local access roles and local conservation roles. And finally, what we all know, which is that access and benefit sharing policies are coming along, but there's still some further steps that need to be done to really balance the needs of plant breeders and formal seed systems, and also the needs and rights of farmers to maintain and exchange their diversity. In situ conservation, on-farm conservation really still needs to be embraced much further if this diversity is continue to evolve in these places alongside climate and pests and diseases, and if the evidence and our belief that this diversity is not only important for the formal seed system, but also important for local autonomy, resilience, productivity. If we're going to embrace that, then further efforts really need to be done to support that side. Approaches still need further innovation. That said, there are a lot of approaches now going on supporting farmers and communities in their efforts in recognizing that diversity and documenting that diversity and increasing demand for local diversity. And one sort of conceptual burden that we still hold after 50 years is that we're still trying to conserve the land races in those places, and that is challenging because those land races are naturally always changing. And so the conceptual change really needs to be a focus on conserving or embracing the conditions and processes which support that diversity to evolve and change as needed, rather than focusing on particular diversity itself. There are interventions that external actors can do to support those processes. We provide a number of ideas in the paper. Happy to talk about that more. For crop our relatives, I just want to emphasize again that it's really, the literature is not good. We're really losing a lot of populations around the world very quickly. And so there are also methodologies that have been invented and understood on how to try to protect them. They're not just for crop our relatives, it's essentially what's done for wild plants around the world. And those need to be ramped up essentially. Informal systems, seed systems, finally. Continued advocacy for diversification of the genetic basis of commodity crops is probably important to avoid major production losses. We still really don't know. And I think this homogeneity story really needs to be unpacked further to know where we're at with genetic vulnerability. In essence, one of the things I want to say is that this has been a study of crop genetic erosion. We still really need a study of crop genetic vulnerability. The last big one that was done was 50 years ago, the National Academy of Sciences study in the United States. Reinvestment in public breeding programs is, and that's pre-breeding and diversification sort of work, is likely to be critical if we're really going to be able to diversify or support diversified bases. At least here in the United States, that public breeding has been declining over decades. There is really interesting farmer participatory work with modern cultivars and formal seed systems, particularly in Europe, and that is very interesting. And I think that we need further critical assessments of the major trends going on, like industry consolidation, IPR, and really understanding how that is affecting crop diversity and how to mitigate the negative effects on diversity. My final slide, after I show you this beautiful picture. So all of what I just mentioned in terms of actions that we can do is really just about mitigating, or perhaps if we're lucky enough, stemming loss of crop diversity. The third aspect, actually reversing this loss and going to more diversified systems, again, globally, is really another whole picture entirely, in which I think probably nobody knows the whole story of how to do that. Certainly I don't. What we're looking towards, I think, is kind of known, which is global agriculture and food systems that are diversity supportive, that continue that diversity and don't essentially narrow it over time as its major process. That means more species varietal and genetic diversity in agricultural systems. And it likely also means more local autonomy and market supporting processes. How to get there, though, I think is really anybody's guess. And I'm sure the well over 100 participants on this talk would have well over 100 different ideas about it. One certainly clear aspect to me is that education and awareness, societal recognition that crop diversity underpins our survival is going to be part of that. And therefore, we do need to continue to ramp up our public education around how precious this diversity is to everybody. I really appreciate your time. I really hope that this was useful for you. This paper is online and available, open access for free right now. Please look at the supplementaries for particularly the gene bank information, and really happy to stick around for a couple of minutes and have any conversations you'd like to have.
Thank you, Colin. That was excellent. Thanks very much for that overview. We have about 14 to 15 questions. So what I would suggest is maybe we do a little bit of a rapid fire, maybe you, so we can get as much as possible as feedback that we can to the people that are here. So I'll start out with a very interesting one. Many countries advocate cluster farming as a means to reconcile food shortages. And by cluster farming, they mean they organize the farmers that are close together with the same type of crops and everything so they can manage it a little bit better. What can you share to us or say to that effect? Do you think that you oppose that? Would you oppose that because of a loss of diversity, or would you be in agreement with something like cluster farming?
Yeah. So rapid fire means I answer this as quickly as possible. So I wanted to, well, one thing I wanted to say is that there's a couple of interesting papers that have tried to look at crop diversity, let's say change at sort of that level of spatial scale, and have seen consolidation into these specialized areas, for instance, counties in the United States, as a trend that may be worrying, maybe not, but it is seen and talked about, and you can find those papers in our article if you're interested. I don't particularly think it's an issue until it's known to impact a vulnerability. And that would really have to do with how many varieties are being grown in that place and how genetically similar they are. And I think intuitively, but really, again, we really just need more research to really understand this. We have the few cases like the Southern corn leaf blight disaster in the 70s that are indicative, but really understanding in these cases when you get to a point of uniformity where it's really concerning. And I think that is essentially still very much unknown, and probably crop specific and even trait specific dependent.
Yes, good, thanks. This one's from Desterio Nyamongo. Ex situ conservation removes material from the environment and arrests the evolutionary process. With the accelerated climate change, what are the chances that we could be holding material in ex situ conservation that may not survive the pressure of the environmental conditions 100 years down the line?
I think that it's 100% likely that some of the material in gene banks, if you plant it out now, won't thrive very well, for sure. That said, I think it's also 100% likely that some of it will, because there's such a tremendous variation within there. And of course, we're not only interested in just planting out what's in gene banks, but also in using the genes themselves to breed in. So it's a complicated story, but I think underlying that question is an important point, which is that we need to not just rely on ex situ systems, but on the continued evolution in situ. And that does mean embracing somehow these on-farm or in situ systems to some degree.
Could you please elaborate on how loss of genotypes not necessarily implies decline in diversity?
Yeah. Yeah. Um, yeah, that's a complicated one to do rapidly, but thank you for that question, because I know that I went very quickly through that. And so loss of genotypes in any given area certainly do mean a loss of things. But the complication is that very often, if you look at the whole system, whatever's going on, there's also been an introduction of other diversity. And so if measurements happen, which they very often do in these studies, across a whole bunch of varieties, for instance, and looking at total neutral diversity, in many cases, you won't see overall loss. You'll see maintenance or even an increase in diversity because new things have come in, even as some things have been lost. And so it's really a matter of focus. Are you concerned about particular things that are really valuable and special, specific things being lost? Well, then, yes, there's a real worry there. If you're worried about overall neutral diversity in a certain place, then you might not be as concerned. And you might be worried about both of those things. And I think that's probably the way to go as well.
A little more general, what role do you think the CG system has played in diversity shift?
Yeah, I highly invite folks to read the recent article from this last year from Gatto et al. 2021 that has looked at the rollout in the developing world of essentially modern varieties, especially these mega varieties, largely by the CGIAR across 11 crops. And it's been very successful in establishing, of course, modern varieties in many regions. What has been the impact on overall diversity is very complicated, I think. But I think intuitively, it's very likely that if singular mega varieties are taking over large spaces of land, that there has been an overall decrease in situ diversity through CG practices. And I'm a CG person, or I count myself as one of those. What is the relevance of that in terms of local vulnerability or susceptibility? I think is really not always known, although there is some evidence there that can be troubling in some places and fine in other places. What is the over impact globally is a whole other story, and it really has to do then with gene banks and their role and our ability to quickly move diversity when needed. I apologize, that's a very quick answer to that one.
Fine, that's okay, we understand. When talking about genetic erosion, shouldn't we add the diversity in gene banks to the diversity in the fields to know if something is being lost? Could it be that with the limited loss observed in the papers that you analyze, it's compensated by the wealth of diversity conserved and available in gene banks?
I cannot wait until a sophisticated study comes along that really encapsulates both sides of that story and across multiple crops in a region. There are some studies that try to do both sides of that story for a specific crop. Again, really take a look at that McLean Rodriguez et al. from these last couple of years. Just really beautiful work, beautiful PhD. But yeah, we need that bigger picture, and that's going to be really hard to do, I think. But it's almost like we've been working for a couple of decades on getting ourselves together with the computer technology, the spatial stuff, the occurrence information, attaching that occurrence information to passport data and gene banks. We kind of set up for this. And I think in maybe a decade from now, we'll really know how to do this more holistic stuff really well, maybe faster when the smarter younger generation attaches themselves to this idea. We need to sell that need.
Yeah. Here's another very interesting one about collecting. Continuing genetic dynamics in land races and wild relative populations means that we should keep collecting the existing and emergent diversity. Nonetheless, some gene banks have essentially stopped further collection missions. ¿Qué pasó is the question. What happened? Why aren't we collecting more? Why aren't there more collecting initiatives worldwide?
Yeah. It's rare to find a practitioner in our field that feels that there's no further need for collecting and conserving. On the other hand, it's very common to find protections in our field that are constrained by at least two major issues. One is just funding, and they simply can't do more. And they recognize that further collecting means more work, more efforts, especially for wild relatives and things like that are more difficult. The other aspect, of course, is the politics and the changes that have constrained this collecting, especially international collecting in many regions for the last 30 years now. There is hope, of course, on that side, on that political side, that things are improving. And there are some wonderful global projects that have really pushed that edge and shown that international collaborations are still possible. I'm an optimist, I suppose, and hopeful. I'm hopeful that the politics are going to improve, and also hopeful that gene banks will be better sourced in the future and not less. But I realize that many of you might sigh because you've seen declines mostly in your careers. And I feel that pain.
This has to do with regeneration. And it has to do with the graph that you showed with the decline in diversity due to regeneration cycles. Gene banks regenerate or multiply their original plant material for multiple times due to viability loss or small stock. Can we say that the original plant material is the same after the regeneration? What is your recommendation in context of regeneration frequency and genetic erosion?
Yeah, thanks. Yeah, mostly what I want to acknowledge here is that there is a good set of literature that really looks at this and also really tries to advance the science on the best balance between number of regenerations and maintaining diversity, because it is highly likely that through every regeneration there's going to be a little bit of a change or a little bit of loss of diversity. And so I mostly invite you to look at that literature, because I'm certainly not an expert on that, but there are many experts out there and folks doing new modeling for other types of species that we don't know as much about in the past. For instance, check out Sean Hoban, Hoban, for a lot of new research on that side, especially for wild plants. I wanted to mention that there's a really neat paper, which I'm forgetting the author, but I can somehow send it to the panelists later, that looked at two different gene bank, the same exact accessions collected in a country, but two different gene bank systems, and how they maintained over time in slightly different ways that diversity. And it turned out that one of the gene banks was able to maintain the diversity pretty well, the other gene bank less well. And that's quite an interesting paper to unpack a little bit what might be those conditions. I will say that the gene bank that was able to do it was likely better resourced financially than the other one, which is unsurprising.
Not as much. Yeah. Thank you. This person congratulates you on your talk and asks, I'm wondering about the relationship between linguistic evidence and genetic diversity. Did you find any evidence of the increased name diversity, and was it well correlated with functional or genetic diversity among land races?
Mm-hmm. Yeah. We certainly acknowledge in our paper that name diversity is out there, and it's been one of the ways that we've tried to understand this diversity for a couple of reasons. It's around in papers and such, but also that's a major way by which we can talk with farmers and try to understand this diversity. We also acknowledge that way of trying to understand change has its challenges. And it turns out that in the literature, one can find both evidence that there actually is a pretty good alignment in some cases between names and genetic diversity, and then a whole other set of literature, which I think is a more abundant literature, that will find that the correlation between names and genetic diversity is poor. And so I think that is probably what the questioner is asking about it. I think it's often the case that it's not the best metric if there are other ways to do it, but it's a metric that is really feasible and possible in many places and probably will continue to be used.
Okay. From Clarice Coyne in USDA, do any of the studies examine or include Asia? She notes the persistence of land races in China into 2010.
Yeah, definitely look in. in the supplementary materials, we provide the entire analysis in a big table, and you can sort just for regions such as Asia or for specific countries you're interested in, and just find those papers. So please feel free to look.
Okay. From Tashome, is there no decline in genetic diversity? Can we say cultivars were lost? If there is no decline, were any cultivars lost? Isn't breeding aiming for an incremental trait genetic benefit, and cultivars should change when they get tired anyways? Was variety replacement factored into cultivar loss?
Yep.
Several questions packed into one.
Yeah, what a fabulous question, really. I think because that is an acknowledgement that agricultural diversity systems do change, and this idea that we should keep them completely static, except in ex situ repositories. I'm talking about out in the field or in seed systems, that we should keep them static is probably a lost cause. I mean, it doesn't make much sense. And in fact, if we want and then we embrace innovation, then of course change is going to happen. Yeah, absolutely. And that is why, you know, there's something to be said for these papers that just look at overall diversity across a lot of different varieties and try to see if the overall diversity has declined over time or not. It turns out that in many cases it's not declined over time, and so that's exciting. But I do want to note two things. Again, one is that this increasing homogeneity does seem to be happening even with increasing diversity, and that homogeneity may be fine, but if it's in key traits, like with the southern corn leaf disaster, that key cytoplasmic male sterility trait, then we might get in the territory of genetic vulnerability. The other thing that I simply wanted to mention is that it would be really nice to feel more confidence in overall diversity non-change if we knew more about functional traits, not just about neutral traits. And so if we can get further on there, then I'll feel more confident that we're doing okay.
Okay. Do you think that whatever the diversity has been conserved in the gene banks truly represents the diversity available in a particular crop species?
Well, okay, so if the question is whether what we've got in gene banks represents what was ever once out there in the world, then I think no, I don't think we probably have anywhere near that for most crops, with maybe a few exceptions. The key word there, though, that what was said was available. And so if the question is, have we got what's in the gene bank versus what's now extant, what's still living and could be collected? I do think actually for a couple of the most sort of key crop, like biggest crops like rice, we might be close to having most of the great majority of the diversity that's still around out there that hasn't already been lost. But in most other crops, there's still a lot of work to go. That doesn't mean, whoever's asking this, that doesn't mean that you haven't done a great job in your own region for collecting the diversity of under your mandate. I'm sure there's many people that have really completed their jobs, but the global picture, I think, is rarely completed.
This question from Ali is how to deal with ex situ conservation of crop wild relatives. He notes that conserving crop wild relatives is more challenging in conservation than other regular crops. Best practices for conservation in ex situ should be established, especially in developing countries. So it's more a comment about the challenges of crop wild relatives and there being no specific guidelines or maybe financial help to conserve them in developing countries.
That's just well noted. And I simply want to add that there is ever evolving good science that have providing indications about how to do this. But I do think that science is also showing that it's hard to generalize. And so there is, again, a paper, I mentioned a researcher named Hoban who studied about 6 different wild plant genera and tried to see if what was needed for some of those genera could be used in other genera to do the conservation well. And it turns out that it wasn't very easy to generalize. And what that means is that we need specific knowledge. And as much as recommendations are going to come from FAO or from Biodiversity International or that, I think it actually in many cases is going to come down to uh, either yourself or particular folks who know a lot about the biology of that particular species. If you can find those botanists or geneticists that know about that species and work with them.
Okay. I think we're winding down now. I'll give you a chance to cut us off whenever, if you're tired now, Colin. We have a really interesting question about policy from Namibia. In terms of collecting and distributing plant genetic resources from the ex situ collections, how do you address the issue when countries do not provide their national collections, but do allow new collections to be brought in from other external collections outside of that country? So there's that the issue that they do not want to share, but they do want to receive, maybe through the international treaty. What would you say to that? Is the question?
Yeah, if I'm understanding that the question is how to improve that system or make it somehow better, it's a great question. I would, because this is a rapid fire question, again, because this is a great like 1 hour answer question, I would simply say that I really acknowledge that there are real reasons why folks around the world have felt that others have taken advantage of them and of their diversity. That is real and needs to be appreciated and respected. On the other hand, there is a lot on us scientists really do understand that that the open exchange of diversity is incredibly important for the advancement of research and science and of food security. And so both of those realities really exist, and neither of them is going to go away. And so I think the question is sort of how do we get from here to the next level? And we're in a period of time with the convention and the treaty that feels troubled. It feels like things could go almost any direction, and they could almost fall apart, and at some levels, and that worries me, because I do think that somehow keeping up that side, which maybe is us, the scientists, in part, that side of reminding how much we need each other in food and agriculture and need that exchange, is going to be important as those other forces that kind of want to just kind of dissipate and no longer have these international collaborations are pretty strong right now. How we do it, I mean, I do think we need to demonstrate the importance, and we need to demonstrate better real examples where there's benefits across wider amounts of people, and not just benefits for certain folks. And we need to be really proactive in that. I do think international projects with some money, development money, can be really helpful for that. And so I really hope that we continue to do that work and be involved in that work. I kind of want to apologize again, because I think this answer could really go on longer, and I wish it was more of a discussion, but that's tough.
And it's the same for most questions. I think we'll take one more, and with that, we'll give this a close. The question comes from Hamidou Falalu in Niger, and he says they have been discussing about revisiting the locations where pearl millet was collected at some time in Niger. And they plan to do surveys to understand the main reasons that some accessions were lost. What is your main expectation, or what would you expect if they do get funding and do this work? What would they be seeing coming in Niger to look at pearl millet again?
Yeah,
for maybe decades?
Yeah, I really hope that Dr. Falalu is able to do this research, in part because there is really interesting work that was done in the past in Niger. There were collections done in the 80s, and then collections done around the 2000s. And so this would be a chance to go to some of those same places for the third time over a total time period of about 50 years. And those sort of data sets are rare to be able to do, and would provide a lot of incredible knowledge and be fascinating. So I really hope you're successful with that personally. What I think would be personally what I would love to see out of that would be work somewhat like the Thorman work that I showed in the very beginning of this presentation, where then after the collecting, if additional work can be done in, for instance, in common gardens to grow all those materials and characterize them at the phenotypic and morphological levels, particularly for traits that are important to the millets, which I'm not an expert in, and also do the genetic analyses, particularly for genetic traits that are known about that are important in pearl millet, which again, I'm not an expert in. But if those things can be done, then I'm absolutely certain that interesting findings will happen. And there's a Vigoro article from 2011 on this that that showed already really interesting change because of climate change in that area. And being right in the Sahel in that area, being exposed to, you know, one of the regions that's really considered the most affected already, I would expect that to advance our knowledge in a real study, this is really happening. This is a real evidence base of what happens after 25 more years of climate change to crop diversity. So I think that's one of the areas I'd focus on. If possible, it would be also fabulous to connect that to the communities there and their nutrition, and whether you can see any of these changes in terms of basically human health and happiness.
Thank you very much, Colin, not only for putting together this incredible presentation and agreeing to come, but also to wade deep into the weeds to try and make sense of all that data. Thank you so much. To all of you attendees, thank you for hanging on with us. It's been great to see you. We're hoping to see you next month. We have a person coming in to talk about knowledge bases and how do you create a plant knowledge base. So we'll hope to see you then. People in the Crop Trust, thank you for your support behind the scenes. Thanks, everybody. Stay safe, and we'll see you next time.
Thanks so much.